Good afternoon, and unusually, I'm the first induced pluripotent stem cell company to be talking in this session, so here's for something slightly different from the morning's talks that you heard already. I'm here to represent the team at BlueRock Therapeutics, which is a wholly owned subsidiary of Bayer Pharmaceutical, and that team and the work that they do and the mission that they represent. So the slides. I want you to walk away from today. You're going to hear a lot of company presentations. I want you to walk away with three things about BlueRock. The first is that we've assembled a team and a talent around a brand new technology that is still in an art form and is trying to industrialize. And so the team that's around that's incredibly important. We have 4 different locations now to our company listed here. And so the first thing I want you to take away is that the organization itself and the culture by which we represent and bring that technology forward. So I want you to walk away with understanding that that's the core of BlueRock's heart. The second thing I want you to walk away with is that we have a very broad pipeline, a diverse pipeline that, unlike many other companies, covers many different disease areas, but at least 7 to 9, depending on the day and how you count them, different cell types and approaches to those diseases. And the last piece I want you to walk away with is that we've built these capabilities end to end. We have the ability to bring brand new academic discoveries like those of our founders in their differentiation processes of cells into the clinic. And now with our lead program in Parkinson's disease, we have the ability to bring those into late-stage clinical trials and are demonstrating and validating that. And last, I want you to approach me. I want to use this forum as a forum to say BlueRock is looking for partnerships. We have a long history of collaborations and several areas that we're looking to collaborate and continue to collaborate in. So what is our mission? What are we doing at BlueRock? I think it's one that's extremely simple and beautiful in its simplicity, but equally as bold as it is beautiful. And what I mean by that is simple. If you think about a Word document where you have the find and replace application within that document, that's exactly how we think of our science. As we age as organisms and as disease comes about, many of the building blocks of our body and our organism, the cells, are either destroyed or simply wiped away. Those cells are gone. And so what we look for is that match between where the cells are the cause or part of the pathology of that disease to our ability to produce that cell at scale and industrialize. And so you'll see that play out over and over again in our pipeline. We have 2 different technologies that we put at play. One is where we build the cell, the very same exact, what we call authentic cell, and bring it into the clinic, and that's our lead assets. And others where, like many others in the room, we use CRISPR technology to edit the genomes of those cells and bring about better therapeutic potentiality of the product. Importantly, at the core and the foundation of our company is the ability to have and make a quality cell type. And the source by which you begin to build an induced pluripotent stem cell is incredibly important. And we've spent a lot of both time and money producing and manufacturing a process by which we can take curated, well-curated material with our partnership with Be The Match, understand the genomes and the integrity of those cells and the history of that healthy individual, and then move them into a proprietary technology that allows us to reprogram those cells back to that naive, very young cell state, the pluripotent stem cell state. We have both the ability to do that and to maintain the commercial rights and all the property that allows us to bring those into the commercial setting. And lastly, what is the foundation is to have many of these cell banks within the freezer that can then be used to edit and go on to a genetically edited cell therapy, or the cell banks that then become the feeders of those differentiation processes that are the final drug product. And what I'm here to tell you is that we've done this over and over again. We have more than we would need in the freezer, and we're beginning to think of BlueRock inside, the ability to take our cell lines and work with partners for those cell lines. And what's very important as you think about and approach the cell therapy field is that not all cells are created equal, and not all iPS clones are created equal. And so careful attention to those cells, their pluripotent state, but also how well they differentiate into the cell type is important to understand and to know. Lastly, I sort of mentioned this already, but I'll use this slide as a way of an example that we look at this as a team sport. It's in our in our birth, in our way that we think about this company, is that this field is too large for any one company to pursue entirely on its own, and we need to lean on each other to be able to make advances for the patients that we're all treating. Good examples of these are listed here. I'll tell you about our relationship with Senti Biosystems, which was a Bayer Leaps venture partner, where we have an ongoing collaboration making engineering genetic elements for the betterment of our cell products, both from locking in phenotypes to as well as kill switches of what you've heard earlier today. Our ability to take academic findings, such as those of our founders at Memorial Sloan Kettering or at UNH, where we use those early innovative cell processes and then begin to both use process development labs as well as into manufacturing at scale for these processes. And we continue to work with academic collaborators as we begin to look at other cell types. So I mentioned the pipeline of BlueRock Therapeutics already once. As you think about our pipeline, I want you to reapply how we go about building this pipeline with that cell-to-disease match. And so what becomes obvious in that very first moment is our lead asset in Parkinson's disease. So the name for this drug, which was DA01, is now bemdaneprocel, and has recently completed its phase 1 enrollment. And I'll spend another slide or two on this important project of ours, but to understand that in Parkinson's disease, when a patient shows up to the clinic, they first complain of either sleep disorder or not speed to gait, the ability to get across or stand up in the room. And at that very first diagnosis with a neurologist, about 80% of the dopaminergic neurons in their brain are gone. So at that moment in time, when you first approach the clinic, first approach a PI, the very cells that give you control of both motor and non-motor function are gone. So there is no drug or small molecule or antibody that can bring those cells back to life. And this is where comes the replace and the restore of that functionality. And so what we do is we deliver dopaminergic neurons to these patients. And now you can go through this broad pipeline and think about that same hypothesis. So as we look at cardiomyocytes, another of the foundational programs that BlueRock was built upon and that our pharmaceutical partner in Bayer has a long legacy within the clinic. These are cardiomyocytes that in the case of a heart function, a misfunction or MI event, those cells are dead. We're not going to bring them back with drugs, so we look to replace those cells. Now, this is bold, this is hard. It's a pumping muscle and a muscle that will squeeze. How do you engraft? Not only that, you need to electronically couple with that muscle as you bring a new young cell type to it. And these are just two of the various challenges that BlueRock has overcome and is now bringing this product forward to the clinic in the near term. You can continue going on immunology. We have both precursor early myeloid cells that we're bringing forward in an oncology setting, as well as early discoveries in trying to head towards Tregs, like many of us in the field, of going after autoimmunity to rebalance the immune system when it's gone sort of out of whack, if you will. And the last is one that I'll use yet again to demonstrate our willingness and our eagerness to partner when we find who we think is the best in the field at producing very complicated cell types, such as RPR or photoreceptors that are used for perceiving light in your eye. We partnered with OPSIS, which is a wholly owned subsidiary of Fujifilm. Now you can think about that collaboration. We have Bayer, BlueRock, Fuji, and OPSIS. One might think this is an impossible endeavor, but I tell you right now, our very next clinical trial that we look to file towards the end of this year, beginning of next, will be with OPSIS in inherited retinal diseases. So every year I come to the meeting on the Mesa or meeting on the Med, and there's always a desire to have me share data with you. I'm going to disappoint you yet again once this year, and I'm unable to share data with you, but I'm excited to tell you that the next time I show up, I absolutely will have data in hand for you, as we plan to share the data from our phase 1 in the second half of this year. And I'm very excited to be able to even call out the time frame for that, which we've publicly told you that we've completed enrollment of June of last year. This is an open label, non-controlled data set of 12 patients, for which we have a low dose and a high dose group. All of those patients have been enrolled, and if you can look back into our press releases, the year prior to that, in May, we had dosed our first patient. So you understand that our endpoints are around 1 year and 2 year for safety, as well as motor and non-motor function within that 1 year and 2 year look. At the very same time that we completed enrollment, from the early data, we recognized that we now have to understand this disease and where our product fits into the disease, which is why we launched a non-interventional study that will act as both a feeder for our late-stage clinical trials, as well as teaching us about the longitude of the disease and the trajectory of those very same patients within that non-interventional study. This is a global non-interventional study looking to enroll about 300 to 500 patients. And it also will allow us to start to understand the endpoints in terms of how we measure those endpoints. So within this study, we will be looking to study digital health technologies, and about a month ago, we announced yet another collaboration with two different forerunners in the digital health technology space. Those would be Emerald and Rune Labs. And so just to give you a little bit of a taste of what these digital health technologies promise to do for us, understanding that as we study Parkinson's disease and have registered several products as an industry within this group, you're really looking at a moment in time for a Parkinson's patients when they show up to the doctor's office or where they record every day in their diaries for their self-reported results. This gives us only a snapshot and is very diverse across the patient setting of how they report or how a doctor reports on this disease. So these two technologies are an attempt to really look at the continuous nature of this disease and how it shows up differently across patients. The first one, Rune Labs, is part of the only FDA-registered device, which is the Apple Watch, that has a motor disorder API within that watch, and Rune has overlaid their technology into this to allow us to study continually for about 50 patients within the United States in our NIS trial for the ability to collect that data both on tremor, on movement disorder, gait speed, as well as sleep. Now the other one, which is Emerald Technologies, is really quite novel within the field, and that is it's an invisible to the patient. And so the patients will have these two technologies, both wearing the Apple Watch as well as the invisible low-frequency radio waves that are put on the wall in their home, and then the algorithm which allows them to follow that patient all around their house while they go about their day. And these two digital health technologies are really looking to see how can we use these to help bring in some of the real-world evidence that will be required of us as we register this product. And then the last piece I really want to leave you with is, again, a call out to those that are in the room that are thinking about different cell types as well. Well beyond the programs that I've told you about, we have an early myeloid progenitor, what we'll call platform. We like to think of this cell type as a cell type that exists as a platform cell type, meaning it has many different indications and many different utilities to it. You've heard the same thing from the earlier speakers around T cells, the ability to create different targeting moieties and use that same cell type over and over again. In our own work, we have now laid the groundwork and demonstrated and will begin to share data with you to validate that we know how to get cells into the central nervous system, that being the brain, and we know how to distribute and engraft those cells to that site. So we look at the first early work of our myeloid progenitors where we think about diseases, and these are just a list of several of them, but you can imagine that complex enzyme replacement in a lysosomal storage disease is an extremely difficult endeavor. Now, we've seen great progress in the delivery of viral vectors, but understand those viral vectors are one at a time. You have to make a different vector for each of the lysosomal storage diseases. These are often ultra-rare diseases, and so no clinical site gets to see these patients over and over again. If you now start to think about our myeloid progenitor cells, these cells make many, if not all, of the enzymes across that lysosomal storage disease spectrum, and you can imagine a single product at that site or for that treating physician that all of their lysosomal storage patients could fit into. So we're excited to bring this forward into the clinic in the near term. You can imagine beyond that, both frontotemporal dementia, which is a disease where progranulin is the basis, again, another enzyme replacement concept. And then finally, you can think of microglia as sort of the homeostatic cleanup crew of the brain. That is that they go around and help create both continuing neuronal connections as well as waste products. And immediately, and some of the literature would really start to support that microglia play a central role in both the pathology of the building plaques in Alzheimer's disease, as well as the role that they play in maintaining homeostasis in an Alzheimer's patient. And so lastly, this is a conversation that we're having now with several different large pharmaceuticals in how to partner this, this asset. As you can imagine, for Bayer and for ourselves, the Parkinson's project is becoming a much larger and bigger endeavor for our company, and so the ability to continue to use our pipeline by partnering those out. So I'll stop here. I'll summarize and just say thank you for your attention. I can see that I'm probably up for time, although I don't see a timer. There it is. And thank you for your attention.